Chapter Cell, Phenomena of Diffusion and Osmosis. So, diffusion, if we want to define the diffusion, how we can define? It is the process of movement of molecules from higher concentration towards the lower concentration. And this phenomena we can understand better with one experiment. In this, in a beaker half filled with water, we have taken a beaker, in that it is filled with clear water, half filled with water. In that if we put a drop of ink, blue colored ink, if we are putting, that ink starts spreading in whole water, and after some time the whole water becomes blue in color, because ink was concentrated and those molecules have been spreaded. The same example we can see that if we are spraying deo at one corner of the room, we can smell in the whole room. That is the phenomena of diffusion. That means movement of molecules from higher concentration towards the lower concentration. That process is called as diffusion. So we can say that movement of gases takes place and it is based on the concentration. Another example we can see, like we have seen in this beaker, and I told about the deos, as well as another example in the respiration process, how oxygen is diffused in the cell and carbon dioxide is coming. Because when we are talking about the lungs, in the lungs oxygen is there, that oxygen have been diffused in the blood vessels, and at the time blood vessels are having more oxygen, that oxygen can be diffused into the cell, because cells are having less oxygen, and cells are having more concentration of carbon dioxide which can enter into the blood vessel than it reach to the lungs. In this way diffusion takes place in our body. When we study the osmosis, other part is osmosis is also a type of diffusion, but that osmosis is diffusion of water. Here we are talking about the gases, here we are talking about the water. Now when we are talking about the osmosis, how it is different from diffusion? In diffusion we said movement of molecules from higher concentration towards the lower concentration. But in case of osmosis, one more word is there, that keyword is semi-permeable membrane, that you have studied in the plasma membrane or cell membrane. So osmosis takes place through semi-permeable membrane. I will define it after this. First try to understand with this experiment. In a beaker, we can take half filled beaker, half water filled beaker, that means in a beaker half filled with water, and in that we can take one small potato and we can take out the cell wall of the potato, that means it is peeled off. So it is having semi-permeable membrane or cell membrane, cell wall have been removed, so that semi-permeable membrane is there, and in that potato we can make a cavity in the center. When we are making a cavity in the center, a potato with cavity in the center, so in that cavity we will fill the sugar syrup, and it is clear from here, outside water is there and inside sugar with water is there. That means the concentration of water molecules are more in the beaker and less in the potato. So what will happen? As I said, movement of molecules from higher concentration towards the lower concentration. So what will happen? These water molecules start moving inside the potato. So when the water molecules are entering into the potato, the level of the sugar syrup starts increasing inside the potato. So we can prove the phenomena of osmosis in this movement of molecules from higher concentration towards the lower concentration through semi-permeable membrane, and this is called as osmosis. To study the phenomena of osmosis in detail, we have to study the concentration of solutions. Types of solution. So before studying the types of solution, first of all you should know what is solution. So when solute is mixed with the solvent, it is called solution. Suppose example we can take in sugar solution. Sugar is the solute, water is the solvent, and sugar syrup is the solution. Now when we are talking about the types of solution, there are three types of solution: hypertonic solution, hypotonic solution, and isotonic solution. Three types of solution are there. So hypertonic solution is what? If surrounding medium has lower concentration of water than the cell. Suppose if I am saying this is one of the beaker, in this beaker water is there. If we are saying if surrounding medium has lower concentration of water, and if I am putting one resin in it, resin is having some cytoplasm inside it, solution is there, and if I am putting in one of the solution which is having more concentration of sugar than resin, then we are saying that surrounding medium has lower concentration of water. And we know that in semi-permeable membrane of the cell, it allows some molecules, that means water molecules, osmosis process we are talking about. So water molecules start moving out of the cell because water concentration is less outside the cell. So we are saying that water molecules start moving out from the cell. Then it is called as cell will lose water, and the solution is called as hypertonic solution. When we come to the hypotonic solution is if surrounding medium has higher water concentration. Suppose we are having resins, and those resins we are keeping inside the water, resins start swelling. So it becomes swell. So that is called as hypotonic solution. Isotonic solution is a type of solution in which there is no net movement. Concentration of water is same inside the cell as well as outside the cell. So we are saying that there are three types of solution in case of solution we are studying: hypertonic, hypotonic, and isotonic.